Morning, good afternoon, good evening. Hi, we are from Group 6 and now we will discuss bioenergetics together with my group mates Colantro, Ofyanga, Ramos, and Belias. So now we will discuss bioenergetics. What is bioenergetics? So it is the branch of biochemistry concerned with the energy expended in the formation and breaking of chemical bonds in biological molecules. Some species, such as autotrophs, may obtain energy from sunshine, which is the photosynthesis, without consuming or breaking down nutrients. Bioenergetics is a discipline of biology that studies how cells convert energy. From the word bioenergetics, bio means life and energetic means study of energy. Biologic systems are essentially isothermic and use chemical energy to power living processes. The way in which an animal obtains suitable fuel from its food to provide this energy is basic to understanding of normal nutrition and metabolism. Death from starvation occurs when available energy reserves are depleted, and certain forms of malnutrition are associated with energy imbalance, which what we call marasmus. Thyroid hormones control the metabolic rate, which is the energy we release, and disease results if they malfunction. Excess storage of surplus energy causes obesity, an increasingly common disease of Western society, which predisposes to many diseases, including cardiovascular disease and diabetes mellitus, type 2, and lowers life expectancy. So now, there are two laws of bioenergetics. So first is the law of thermodynamics. States that the total energy of a system, including its surroundings, remains constant. It implies that within a total system, energy is neither lost nor gained. So now, the second law of thermodynamics states that the total entropy of a system must increase if a process is to occur spontaneously. Entropy is the extent of disorder or randomness of the system and becomes maximum as equilibrium is approached. Under condition of constant temperature and pressure, the relationship between the free energy change of a reacting system and a change in entropy is expressed by the following equation, which combines the two laws of thermodynamics. Metabolism comprises two major parts, which is the catabolism and anabolism. So the first one we will discuss is catabolism. So the prefix kata means to break down or downward, while bolism means is to change. Catabolism is the process of action that set of metabolic pathways that breaks down molecules into smaller units that are either oxidized to release energy or used in other anabolic reactions. It also the part of the metabolic process that breaks down large, complicated molecules into smaller ones in order to produce energy. So the second one is anabolism. So the prefix ana means up, back, or again. So anabolism is a sequence of biochemical reactions involving building complex structures from smaller ones, and they usually consume energy. Anabolism in living organisms includes synthesizing anabolic proteins from amino acid units, glucose from carbon dioxide molecules, and DNA from nucleotides. Anabolic pathways are responsible for synthesizing essential molecules to the life and function of the body. An example of anabolic pathways is the food manufacturing process in plants, where sugar molecules are produced via a pathway known as photosynthesis. Plants consume energy obtained from sunlight to produce sugar molecules from carbon dioxide. Sugar molecules produced by the plant provide a source of energy for animal feeding on these plants. The difference between them is catabolism releases energy, while anabolism requires energy. So now we will proceed in cellular respiration. Now, it is a process cells use to release energy. So now we will proceed in cellular respiration. So now it is the process that cells use to release energy, which is the ATP from food. Usually means sugar, but can also work with fats and proteins and amino acids. There are quite a few chemical reactions that take place in different steps, but overall the idea is pretty simple. Chloroplasts use photosynthesis to convert light energy, water, and carbon dioxide into glucose and oxygen, while mitochondria use glucose and oxygen to produce ATP, carbon dioxide, and water. So now here's a simple illustration. Oxygen plus glucose equals carbon dioxide plus water plus energy. So now that's all.
Hello everyone, my name is Francisca Colandro and I will discuss about the ATP structure and cellular respirations. So now let us first give a little introduction about adenosine triphosphate or ATP. If you guys remember the four major biomolecules in our previous lessons, which is the carbohydrates, lipids, proteins, and nucleic acids. Now, let's define the adenosine triphosphate or ATP. So, ATP, it is a nucleotide. It has a nitrogenous base attached to a sugar molecule with three phosphate groups. So the base is referred as adenine and the sugar referred as ribose and the three phosphate groups. As you can see in my drawing, this is the adenine and this is the ribose and the phosphate groups. These are molecules. Those molecules provide energy for various biochemical processes in our body, such as oxidation and reduction. Anyway guys, the oxidation reductions is a type of chemical that involves a transfer of electrons between two species. The oxidation is the gain of oxygen, while the reduction is the loss of oxygen. Therefore, the adenosine triphosphate is also called energy currency of the cell. These ATP molecules are synthesized by mitochondria. Since the mitochondria use glucose and oxygen to produce adenosine triphosphate or ATP, carbon dioxide, and water. And it also called the powerhouse of the cell. Take note guys that the adenosine triphosphate is the energy carrier. Molecules are found in the cells of all living things, such as prokaryotes and eukaryotes. Adenosine and triphosphate is vital for our cells because it is the primary carrier of energy in our cells since our body has a lot of cells and those cells need energy to function well. By the way guys, in triphosphate it means there are three phosphates in adenosine. Now, let us proceed to adenosine triphosphate structure. As you can see in the picture, it is the molecular structure of adenosine triphosphate. The question is, how does this molecule store energy? The basic structure in storing energy is the adenine, which is connected to the three phosphate groups through a ribose sugar. Then the adenine connected to ribose to create adenosine. The energy stored in the bonds between the three phosphate groups, when this bond breaks down, it releases energy, and that energy are chemical energy, which is carbohydrates, fats, and proteins. The band holding the last phosphate, that one band at the end of the little tail, holds the most energy. It means that little tail has a lot of more energy. That is the phosphate that gets kicked off, and when this happens, it releases a bunch of energy. So, the second question is how does adenosine triphosphate give us energy? So, adenosine triphosphate uses chemical energy obtained from the breakdown of glucose. During hydrolysis, one phosphate molecule is broken off. ATP's tail to release energy. When this happens, when that phosphate molecule is broken down, the the adenosine triphosphate turns into adenosine disphosphate. The hydrolysis, this is the one of the step to break down the bonds of three three phosphate groups. Hydrolysis is any chemical reaction in which a molecule breaks down one or more chemical bonds. So now let us proceed to cellular respiration. So what is cellular respiration? Cellular respiration, it is a metabolic pathway that uses glucose to produce adenosine triphosphate or ATP. When we say metabolic pathways, it is a linked series of chemical reactions occurring within the cell. It is also a series of chemical reactions in the mitochondria where molecules of glucose are broken down to make carbon dioxide, water, and ATP, or adenosine triphosphate. The, the function of cellular respiration is to generate usable ATP triphosphate energy in order to support the other reactions in the body. So, where does cellular respiration occur? Cellular respiration occur in the prokaryotes and eukaryotes. In prokaryotic cell, it occurs in cytoplasm, and in the eukaryotes, it occurs in mitochondria.
For the three stages of aerobic and anaerobic cellular respiration, we first discuss what is the aerobic and anaerobic cellular respiration. In the picture, it describes that the aerobic produce more ATP than anaerobic respiration. Aerobic respiration, it occurs in the presence of the oxygen and in most cells most of the time. It is the process of cellular respiration that takes place to produce energy from the food with the presence of the oxygen. This type of respiration is common in most of the plants, animals, birds, humans, and other mammals. In this process, it has the water and carbon dioxide are as produced as end products. The aerobic respiration with oxygen takes place in the cell's mitochondria, while the anaerobic cellular respiration is the respiratory process that occurs in both prokaryotes and eukaryotes, in which cells break down the sugar molecules to produce energy without the presence of oxygen. It says that anaerobic respiration, which is without oxygen, takes place within the cell cytoplasms. Anaerobic respiration occurs without the oxygens, which is it releases less energy but more quickly than aerobic respiration. Anaerobic respiration in microorganisms is called the fermentation, which will be discussed later. Let's go with the three stages of the anaerobic and anaerobic cellular respiration, which is the first phase is the glycolysis, the second phase is the Krebs cycle, and the last phase was the electron transport chain. Glycolysis. It is the first phase in the cellular transportation or the cellular phase. It is the process in which the glucose is broken down to produce energy. It produces two molecules of pyruvate, ATP, and ADH, and water. This process takes place in the cytoplasms of a cell and does not require oxygen, which conclude that the glycolysis process was anaerobic. During the glycolysis, the glucose sugar molecules, which has the formula C6, H12, and O6, get broken down and converted into two molecules called the pyruvate that has the formula of C3, H4, and O3. Pyruvate, which is called in another term pyruvic acid, is a more usable molecule that helps the process of glycolysis going.
Next, we have the Krebs cycle. Krebs cycle was named after Hans Krebs, who postulated a detailed cycle. It is a very complex process, but it can be summarized in a series of 8-step processes where the acetyl group of acetyl-CoA is oxidized to form two molecules of CO2 or carbon dioxide, and in the process, one ATP is being produced. Reduced high-energy compounds, NADH and FADH2 are also produced. So to start the cycle, the formation of six carbon compounds citrate occurs by the combination of acetyl-CoA and oxaloacetate in the presence of citrate synthase enzyme. The second step, there is the conversion of citrate to its isomer isocitrate in the presence of the enzyme aconitase. Thirdly, the isocitrate converts into alpha-ketoglutarate, a 5-carbon compound prompting the release of a molecule of carbon dioxide. The catalyst of this step is NAD plus and isocitrate dehydrogenase. Then, for the fourth step, the 4-carbon compound succinyl CoA forms under the control of the enzyme alpha-ketoglutarate dehydrogenase. Here, a molecule of carbon dioxide also releases. Also, NADH forms in this step as the enzyme is NAD plus dependent. Fifthly, this step involves the removal of the CoA from the succinyl CoA to form succinate. GDP or guanosine triphosphate forms after the substrate level phosphorylation of GDP or guanosine diphosphate. The catalyst is succinyl CoA synthetase. Similarly, in the sixth step, the succinate converts into fumarate. The enzyme succinate dehydrogenase helps the reaction. The succinate dehydrogenase reduces the FAD to FADH2. Likewise, with the addition of the H2O, the fumarate changes into malate in the seventh step. The presence of fumarase in this step acts as the catalyst. The final step is the formation of the oxaloacetate with the help of enzyme malate dehydrogenase. So after the Krebs cycle loop twice, we will have 2 ATP molecules, 6 NADH, 2 FADH2, and 4 molecules of carbon dioxide will be released. Next, we have the electron transport chain with chemiosmosis. The electron transport chain is a series of proteins and organic molecules found in the inner membrane of the mitochondria. Energy released in this reaction is captured as proton gradient, which is then used to make ATP in a process called chemiosmosis. Together, the electron transport chain and the chemiosmosis make up for the oxidative phosphorylation, which is the last step of cellular respiration, wherein a large number of ATP or adenosine triphosphate is produced. It must be remembered that NADH and FADH2, which are the byproducts of Krebs cycle, are being utilized in this process to create ATP. Like the Krebs cycle, this process is very complex, but it will be discussed in a simpler term to have an easier grasp on it. So for the first part, we have the NADH and FADH2, which are being transferred through the protein complexes and electron carriers. It's these two types of molecule that carry an electrical charge traveling from the mitochondrial matrix through an inner mitochondrial membrane. Both of these molecules are being oxidized, which means they lose an electron in the hydrogen molecule. As a result of the oxidation of the NADH and FADH2, an electrochemical gradient is created, meaning there is a difference in electrical charge between the two sides of the inner mitochondrial membrane. The outside or the exterior of the mitochondrial membrane is positive because of the accumulation of the protons, and inside is negative due to the loss of protons. When there is high concentration of protons on the outside of the mitochondrial membrane, protons are pushed to ATP synthase. This movement of protons causes ATP synthase to spin and bind ADP and phosphate producing ATP. Finally, the ATP is made. So for the end product of this process, we will have 6 water molecules and it will produce a range of 28 to 38 ATP molecules.
Fermentation is an aerobic process in which microorganisms break down organic compounds such as sugars or carbohydrates into simpler molecules to produce energy. It has several noteworthy applications. A. Food and beverage production. Fermentation is used in the production of various foods and beverages. For example, yeast fermentation is employed to make bread rise by producing carbon dioxide gas, and it is also used in brewing beer, making wine, and fermenting dairy products like yogurt and cheese. B. Biofuel production. Ethanol, a type of alcohol, can be produced through fermentation of sugar derived from crops like corn, sugarcane, or cellulosic biomass. Ethanol can be used as a biofuel and is an environmentally friendly alternative to fossil fuels. C. Biotechnology and pharmaceuticals. Fermentation is utilized in the production of numerous biotechnological and pharmaceutical products, including antibiotics like penicillin, insulin for diabetes treatment, and various that is used in industrial processes. ATP is often referred to as the energy currency of living cells because it provides the energy necessary for numerous biological processes. Here are some examples of ATP in action. A. Cellular energy transfer. ATP serves as an energy carrier within cells. When ATP is hydrolyzed, it releases energy that can be used to power cellular activities such as muscle contraction, active transparent of ion across cell membranes, and synthesis of macromolecules like proteins and nucleic acids. B. Metabolism. ATP plays a central role in metabolism, the set of chemical reactions that occur within cells to maintain life. ATP provides the necessary energy for anabolic reaction, building complex molecules, and drives catabolic reactions, breaking down molecules to release energy. C. Signal transduction. ATP is involved in signal transduction pathways, which allow cells to respond to external stimuli. It can phosphorylate proteins, altering their structure and function, thus transmitting signals and regulating various cellular processes. D. Active transport. ATP powers active transport processes across cell membranes, enabling the movement of ions and molecules against their concentration gradients. This is crucial for maintaining cell homeostasis and conducting vital functions like nerve impulse transmission and nutrient uptake.
And that's all for our discussion in bioenergetics, and we hope that you've learned something from our discussion and can be useful in your everyday life. Thank you for listening.